Hi, my name is Brad Hamilton. I'm in the Research and Development Department here at Stemgent. Today we will show you a procedure for how to generate induced pluripotent stem cells from mouse embryonic fibroblasts. This procedure can be used to both generate induced pluripotent stem cells and to study the reprogramming process. So let's get started.
Begin by seeding mouse embryonic fibroblast or MEF cells onto a 15 cm gelatin-coated dish at a density of 4 × 10⁵ cells per dish. Now add 30 milliliters of MEF growth medium and incubate the cells for 2 days at 37 degrees Celsius and 5% carbon dioxide until they are approximately 80% confluent. When finished incubating, aspirate the medium and add 30 milliliters of growth medium supplemented with concentrated lentivirus. Rock the dish gently to ensure even distribution of the medium. Incubate the cells overnight at 37 degrees Celsius and 5% carbon dioxide. With viral transduction complete, let's move on to doxycycline-induced reprogramming. To begin reprogramming, 20 to 24 hours post-transduction, trypsinize the transduced cells and centrifuge them at 200 Gs for 5 minutes. Next, carefully aspirate the media off of the cell pellet and resuspend the cells in growth medium. Once suspended, seed the transduced MEFs at an appropriate concentration.